This is Chemistry of Food Part 2, and in this lesson we are going to look at the structure of lipids and looking at the atoms that make up lipids, how they are different from carbohydrates and proteins. Then we are going to look at the various types of food tests that are available for carbohydrates, proteins and lipids. And for your exam, you will need to know the observable changes that take place when you apply specific tests for carbohydrates, proteins and lipids. Now we will look at some key points for lipids. So the first point is, in terms of our diet, we ingest lipids, or lipids are important for our diet because they are the most efficient energy store. So lipids are the most efficient energy store. Okay, so we store energy in the form of lipids because that's the most efficient way for us to store energy. Furthermore, they do provide us with an important source of energy. So, as well as storing energy, also lipids are an important source of energy. Okay, so they are forming two vital functions here. One to store energy, and also one to store energy and one as an important source of energy as well. Again, when we think of it in terms of a whole biological organism or at the system level, we can say that lipids are important for building of hormones, okay, so for hormones to be produced correctly, we need lipids. They're important for the nervous system. Okay, so we've got building of hormones and vital for nervous system. This again makes lipids very important for communication within the body because hormones are chemical messengers and the nervous system is electrical messaging. So for communication within the body, lipids are important. Another important aspect in terms of the system level is that lipids are crucial components of the cell membrane, specifically the phospholipid bilayer of the cell surface membrane, which controls substances going in and out of cells. So, important for structure of cell membrane. Now, the reason they are important for the structure of the cell membrane is because they make up, lipids help build up the phospholipid bilayer of the cell membrane, which regulates substances coming in and out of the cell. Now at GCSE you do not need to know the phospholipid layer in detail, but if you do biology to A level, then you will be taking apart this phospholipid bilayer in more detail and looking at how lipids play a vital role in constructing the cell membrane. So these are just a few of the components, let's say at the system level. So one, it's an energy store. So for a living organism, needs to store energy, and the most efficient way is to do it via lipids. Another important point in terms of energy is that lipids are a good source of energy for living organisms. Then if we also stay at the system levels, we've got building of hormones, and then we've got vital for nervous system function, and also for the structure of cell membrane. Now we're going to look at lipids at the molecular level and look at some of their properties there. And you do need to know lipids at GCSE level, at the molecular level, and know specific details of that. So this here is just a quick summary of lipids at the, let's say, system level or organism level. And now we're going to look at the molecular level of lipids and how though that structure helps in its key functions. So before we go on to the molecular structure of lipids, I just realized that there were two more important points of lipids that I had left out from the previous slides. So these two roles of lipids are important. They are a way in which animals insulate themselves. So insulation for animals, okay, and they can also be used for insulating key vital organs. So also important to insulate key vital organs. Okay, so insulation for animals. So this can be seen in terms of just certain animals have a thicker layer of fat, or sometimes they're known as a specific layer of lipids, such as for whales and let's say seals, they have a certain thickness to their insulation layer, which is known as blubber. But aside from that, all animals have fat for insulation purposes, and also this is important when we talk about key vital organs that need insulation. So insulation for animals. Another important point is that it keeps, let's say, animals that come into contact with water for long periods of time, they need to be kept dry, because if their core body temperature falls below a certain amount, then they can go into shock. And this can happen if the animal is, let's say, exposed to the water, and its body temperature will drop. So to keep the animal dry, certain aquatic birds and animals have a property by which they have water repelling features, either on their feathers or on their fur or on their body. Now this keeps them dry to a certain extent and helps them survive. So what we say is to keep aquatic birds and, let's say, mammals dry. Okay, due to water repelling feature of lipids. Lipids have a hydrophobic component, i.e. meaning that it will repel water. So when we come on to now listing, when we talk about lipids at the structural level, molecular structural level, I will mention this word hydrophobic again. The first atom we're seeing here is the carbon atom. Then we see an oxygen atom. And then finally we will see the hydrogen atom. So the one in green is your oxygen, and now we have the third atom which makes up lipids, which is the hydrogen atom. So could you just put down on your diagram that lipids are made up of carbon, oxygen, and hydrogen. Now putting the carbon, oxygen, and hydrogen atoms together in a specific way, we can make up our lipids. Now, lipids are made up of two essential components. One is called a glycerol and the other is a fatty acid. So, can you take this down? Lipids are constructed of two main components. Okay, so they're constructed of two main components. One is, we call it a glycerol. Let's not put it A. One is called a glycerol. Okay, and this is essentially made up of an organic compound and is 3 carbons in length, 3 carbon chain length. So it's simply made up of an organic compound. You just have to understand it's an organic compound and it's 3 carbons long. And when I say 3 carbons long, I mean it's the chain length is 3 carbons long, and this will become in the relevance of this 3 carbon chain length will become much more clearer in a minute. So it's 3 carbon chain length. Okay, so it's an organic glycerol, it's an organic compound, and it's constructed of 3 carbon chain length. Okay, now the next part of lipids is, let's put it in red. The next component of the lipid is known as the fatty acid. Okay, and the fatty acid are described as being long chain hydrocarbons with a carboxyl acid group at the end. So fatty acids are long chain hydrocarbons Okay, long chain hydrocarbons. Notice this is different from the glycerol, which is only 3 carbons long. So fatty acids are long chain hydrocarbons, and their long chain hydrocarbons are said to be hydrophobic, meaning that they do not like water. Okay, so the long chain hydrocarbon part of lipids in the fatty acid is what gives rise to its hydrophobic property. So long chain hydrocarbons, and at one end of this long chain hydrocarbon of each fatty acid, there is a carboxyl acid group. At one end there is a carboxyl acid group. We will come on to carboxylic acids later, because they're a specific functional group that we look at in organic chemistry, but this is in biology. But you're coming across something known as a carboxyl acid group, and it's found at one end of a fatty acid molecule. Okay, so carboxyl, so let's just write, okay, so if you had to put the formula of a carboxyl acid group, you can see it here. So this is a bracket, by the way. It's a bracket dash carbon, oxygen, oxygen, hydrogen, and close bracket. So this in essence is what makes up a lipid, so any type of lipid. And generally we are going to be looking at a specific type of lipid known as a triglyceride, and there's a clue in the answer as to how these types of lipids are constructed, triglyceride. So that's what we're going to look at now in a bit more detail, about how you should be aware of lipids for your exam. Glycerol component, and generally glycerol, you can see it represented in yellow in most of the diagrams, but here a shading has been given in a different color to indicate the glycerol component that stays intact. Now this is the glycerol before it's attached to the 3 fatty acids, forming your triglyceride. So here you can see the 3 carbons, so you can see 1, 2, 3. Okay, and going off each carbon we can see an OH group. Okay, so we can see an OH group coming off each carbon. Now this is important because these OHs are removed in a type of reaction which you don't need to know at this level, but again, if you do A-level biology, you will need to know these specific reactions. But these 3 OHs are removed and a fatty acid chain or tail will attach to each position here indicated. So this is your glycerol part of your lipid. Okay, so just make a note of this, and this is glycerol before being attached to a fatty acid. So just make a note of that. So if you take this diagram down, make a note, glycerol before being attached to a fatty acid. Okay, I'm going to show you how this molecule looks in total when the reaction has taken place and we have our triglyceride, but we'll just go in steps here. So this is glycerol, and you can see the 3 carbons, and you can see where the OHs are indicated. That is where a fatty acid will be substituted in each position. See the fatty acid molecules. Now notice you have got the acid head. Okay, so this gives the name because of this group here. This is how we get the acid part of the molecule. This is why the molecule is called a fatty acid, because of this group here. Okay, which is, and it wasn't clear before, so I'll just make it a bit more clearer here. Because of this group, it is this functional group, okay, the carboxyl acid group. Because of that, you get the acid name to the molecule, and you can see it here. It's represented here and here on the molecule. Okay, so that's that part of the molecule. So let's just take it off so it's a bit more clear. Now you can see the long hydrocarbon tail, and notice the word hydrophobic. So this chain gives a hydrophobic property to the fatty acid or lipids in general. And you may notice 2 more words here: saturated and unsaturated fatty acid. Now I want you just to pause the video here and think about, or type back on Teams, why we have a saturated fatty acid and then another molecule labeled as an unsaturated fatty acid. what could be the difference? What structural difference are we seeing that gives rise to these two different names? Okay, the key point here is the carbon to carbon double bonds that you're seeing. So these double bonds that you're seeing here, they give the property of saturation or unsaturation for the fatty acid. It's not the double bond at the acid head part. So it's not this double bond. It's not that double bond. It's the double bond within the hydrophobic hydrocarbon tail that makes a lipid saturated or unsaturated. And if it's got a double bond, it's called an unsaturated fatty acid. If there are no double bonds, it's called a saturated fatty acid. And this has huge implications in terms of the structure of the final lipid, whether it's also going to be a solid or a liquid, but also it has huge implications for our health. And we'll come on to talking about this later, but I want you to think about which fats do you think are more healthy. And you may have heard this in advertisements when they're advertising certain type of foods. A certain type of fat is always mentioned. So which type of fat, just think about it as we move forward, which type of fat is more healthier? And it's either going to be saturated or unsaturated. And I want you to think about that as we move forward. So what I want you to do is take the diagrams down for just a simple saturated fatty acid and an unsaturated fatty acid. What I will say is you don't have to make your diagram exactly this hydrocarbon chain length. So here you've got approximately 13 carbons on this saturated fatty acid. You could very easily just make that around 6 if you wanted and make that your fatty acid. You don't have to do the entire chain length, because one important point about fatty acids is that the carbon chain length, the hydrocarbon tail, can vary in length, and this also will affect the final structure of the lipid. So can you just take down a simple drawing of a fatty acid? What is important is the acid head must be there, okay, and then you have a hydrocarbon tail, and you can make that maybe just, I would say, even 5 to 6 carbon long. That is fine. So you can do one saturated fatty acid, and then can you draw one unsaturated fatty acid? Again, just truncate the molecule, make it a bit smaller, and just include a double bond somewhere, and then indicate it's unsaturated due to the double bond. So this is how a fatty acid looks. Now we need to see how we combine a fatty acid to a glycerol to give a triglyceride. Here. And I want you to type the answer back to me in Teams so I can just quickly check that you are following this through. We have two molecules here, and we're going to label this one as A, and we'll label this one as B. I want you to tell me or identify the unsaturated fatty acid and the saturated fatty acid. So you choose which one is which, and I would like an answer back on Teams. So look at the molecules carefully and quickly, just a quick feedback question here. Just tell me which one is the saturated fatty acid and which one is the unsaturated fatty acid. Make our triglyceride. So we are going to combine these 3 fatty acids you see here on the right hand side with the glycerol. Now what I want you to observe as I bring each fatty acid close to the glycerol. So let's take our first fatty acid. Now look where I'm lining up this fatty acid molecule. Okay, so I'm lining it up there. That's the first one. Let's take the next one. And let's line it up there. Okay, and then we'll take our final, is it this one? Just check, yeah, it's this one here. And we line it up here. So we've got our three fatty acids chained, and notice where they are lining up against. Look at the molecule, look at the atoms that they're lining up with to form the bond. So I'm highlighting it so you can see it here. Okay, it's not catching it perfectly, but it's the OHs. So it's lining up with the OHs on each one. I'll just do it here for reference. So that repeats itself for each fatty acid as it binds. So as the fatty acid joins the glycerol, it's joining at these points here. I want you to pause the video here. You don't have to draw this molecule just yet, but I want you to think about what type of reaction is going to occur for these molecules to combine. So for the glycerol to combine to each fatty acid at these specified points. Okay, so let's just try to make that a bit more clearer. Let's just try again. Okay, it's not doing. Okay, so at these points where the glycerol is joining the fatty acid, we are going to get a specific type of bond. Okay, and I'm going to say the reaction is called a condensation reaction. So what could be given out, and what resulting bond do you think we are going to end up with? So just think about this. Maybe you can sketch the answer down on your piece of paper as you're looking at this, but pause the video here, have a think about it, take maybe 30 seconds, take a minute, think about it. I'm telling you, this is a condensation reaction. Look at the atoms involved, and what do you think is given out if this is a condensation reaction? Don't worry about the exact bond being formed just yet. You can actually piece that together as well, because if you know it's a condensation reaction, you know what's going to be given out, so you can sort of do an educated guess in terms of what type of bond is going to be left between the glycerol and the fatty acid. So just think about this. So can you jot down what do you think comes out when each fatty acid joins the glycerol, and what type of bond do you think is formed between the glycerol and the fatty acid? And the clue is that this reaction is known as a condensation reaction. Here is the answer. As you can see here, the three fatty acid molecules combine with the glycerol via the condensation reaction, and water, 3 H2O molecules are ejected, and we get a triglyceride molecule, and you can see here that the oxygen bond is between the glycerol and the actual fatty acid molecules. Now the question may arise is which hydrogen was joined to form our water molecules. You don't need to know in detail at this stage which hydrogen it was. However, we will look at something similar to this when we look at organic chemistry and we talk about electronegativity and the electronegativity of an oxygen and a double bond, and how that subsequently impacts the ability of a bond to be held. So the bonding between oxygen and hydrogen here, for example, and here is not going to be the same due to the electronegative pull of oxygen from the fatty acid and also the presence of the double bond. You don't need to know in detail about that, but what you can sort of appreciate here at this level is that the glycerol combined with the fatty acid molecule, they join via condensation reaction and you get a triglyceride molecule. And lipids, at what you need to remember is lipids look in this manner, with a glyceride, with a glycerol molecule and 3 fatty acid molecules combined to it. A more simplified version of our triglyceride, and you can see here how the glycerol and the fatty acids are represented and how they are combined together. And at the very least for your exam you must know this diagram here. So you must know for your exam that lipids are represented via a glycerol and 3 fatty acid molecules joined. Okay, and if you look at the key as well for reference, they are joined via an ester bond. Again, that's a bit more detail than you need. However, at the very least you must take away from this lesson is that lipids are made up of a glycerol molecule and 3 fatty acids. So you do not have to take the specific bonds mentioned, the molecular structure mentioned in the above diagram. You can if you want to, because it will help you in a few months when you do when you come on to A-level biology. It will help you and you will already have this understanding. However, you can keep it at this diagram here, and please note that your lipids are made up of glycerol and 3 fatty acids. Quickly summarize the lipid content part. So lipids are fats, okay, and these fats are solids, okay, and they can also be oils, and these oils are liquids. Lipids are made up of one glycerol molecule, plus 3 fatty acids. They can be thought of as being saturated. This means no double bonds. Okay, and saturated fats are generally found in animal fats. And also tend to be, in this instance, saturated fats are considered unhealthy. And then you've got unsaturated fats. Unsaturated fats. Let's put fats here. Okay, and these have double bonds. Okay, so you find the double bonds in the fatty acid molecule. So they have double bonds and they tend to be plant oils, e.g. olive oil. Okay, and you can go further with unsaturated fats. If there is more than one double bond, we call it a polyunsaturated fat. So if there is just one double bond, it's called a monosaturated fat. And if you go further, you can get polyunsaturated fats. And you would have heard that again in certain advertisements for certain products. Polyunsaturated fats, good for your health. And So this is in terms of saturation and double bonds. Now, the other point, another few points that I want you to take down for lipids is that lipids are in general now, so regardless of whether they are saturated or unsaturated, lipids in general, lipids are molecules that are insoluble in water. So you need to know a general property of lipids due to their, you should be able to understand now why they would be insoluble in water. They have a hydrophobic section, which if you refer back to the video where we looked at the fatty acid molecule, the long hydrocarbon was our hydrophobic component of lipids. So this is where we get the insoluble nature of our lipids. Also, what we can say is that, okay, so what we'll say is that they have a hydrophobic property. Hydrophobic property. Okay, and the hydrophobic property can be seen in the fatty acid molecule component. Going to move on to food tests, and we are going to look at standard food tests for carbohydrates, proteins and lipids. Before we do that, I'm just showing you a summary of carbohydrates, proteins and lipids. So here you can see the corresponding function of carbohydrates, proteins and lipids, and then there's another column to show you where you can get these three types of food categories in the day-to-day items that you can consume. And you do need to know at least a couple of sources for carbohydrates, proteins and lipids. So take a look at this table. If you're happy with all the knowledge that's been stated here, all the bits of information that has been stated here, that's fine. You don't need to take anything down further. But if there's anything in this table that seems new to you in terms of where you can find carbohydrates, proteins and lipids, make a note of it and make sure it's clear. Alright, okay, so let's move on now. So I'm going to go over certain food tests, and we're going to go through each one, and then I'm going to show you a diagram with a bit more information regarding the food tests, and I'll talk about it. So can we put the subheading carbohydrates? Okay, so we're going to see two tests that we can take for carbohydrates. Right, so the first one is called the iodine test. So the first test that we come across is known as the iodine test. Now this test will test for the presence of starch. So iodine test is used to see if starch is present. Okay, so this is what the iodine test is used for. So we have, this is the name of the test, and what you're testing for is starch. Okay, so could you just quickly make a note of that? The first test is known as the iodine test, and we test for starch. Okay, now what is the result of the iodine test? So what is the observable result of the iodine test? Now, I'm just going to put this in another color. So the iodine test, what will happen is that, let's put it in orange. Yellow, let's say yellow or red iodine. So yellow or red iodine, okay, will turn, let's put this in white, okay, turns, okay, let's say this in a slightly different way. Let's say, let's put that there. Yellow or red iodine solution, okay, I'm color coordinating it so it's easier to remember for you, and you can maybe match it a similar color when you write it in your notes. So I'm saying yellow or red iodine solution turns blue black. So let's take a darkish blue. Let's take, it's not going to be easy to see. So let's say turns blue black. Okay. Turns blue black if starch is present. Okay, so in the presence of starch, iodine solution, which is yellow red, will turn blue black. All right. Now what's important here is that one, you know the reagents, you know that it is iodine, and you are going to be testing it on a specific carbohydrate, which is starch. So you're going to test for the presence of starch. You need to know that the iodine is a yellow or red in appearance, and if starch is present, the iodine will turn blue black. So a positive test for the carbohydrate starch is blue black. All right, so let's look at this in terms of a diagram. The test for starch. Now it's been carried out on a potato, portion of potato, to show for the presence of starch, which is a storage molecule. So the potato plant will form starch, and it will be stored, because it's a good storage molecule, it will be stored underground. And here you can see starch readily available as a positive test when iodine is added. Now you can do the same test in a solution form as well, where you have starch solution. Here it's in a solid form, the potato, but bear in mind you can have it in a solution form. So iodine solution with starch solution in that form as well. So here you can see that we've got our iodine solution, okay, and this would be, the observable color would be yellow / red, and then here you can see the positive test, which is blue black. Okay, so these are things that you need to remember in terms of the observations for this particular test. Now what's also important here is the hazards, because you do get exam questions that will ask you to state at least 2 safety precautions that need to be considered when carrying out this particular test. So wearing safety goggles, okay, and knowing that iodine solution is an irritant, okay, avoid contact with eyes and with skin and eyes. The other one that's left out here is that it's not really safety in the sense that it can stain skin and clothing. So this is where the avoid contact with skin would come in. Yes, it's an irritant, but it can also stain your skin and clothing. All right, so if you want to just take a note of this, if you understood it from the previous description, that's fine. But here, just take a note of the safety considerations, because that is important. Second test for carbohydrates is known as the Benedict's test. So this is known as the Benedict's test. Okay, and the Benedict's test, okay, can you just put down in brackets, used for, and we can say sugars, but we can be a bit more precise and we can say used for reducing sugars. Okay, so can you just take that down? That this is the Benedict's test. It's the second test for carbohydrates that we need to know, and it's used for reducing sugars. So this test, the reagent starts off, so the Benedict solution starts off blue, and it will turn brick red upon heating if a reducing sugar is present. And an example of a reducing sugar is glucose. And we just write this sentence in terms of what a positive test is for this particular type of carbohydrate. So what we say is blue Benedict's solution okay, turns brick red. So blue Benedict solution turns brick red, and the condition is brick red on heating. So you need to heat the solution of Benedict solution with the sugar, and if the reducing sugar is present, it turns brick red. Okay, so blue Benedict solution turns brick red on heating if a sugar is present. Okay, e.g. glucose. Can you just add to your notes? I've just realized. Can you just add to your note that if a reducing sugar is present, e.g. glucose is an example of a reducing sugar. Now, with this Benedict's test, you can get a varying amount of colors. Sorry, a varying degree of colors can be displayed with the Benedict solution. It's not always brick red. Brick red would be the strongest positive test you can get, but it depends on the concentration of the sugar that you're given or you're testing for. A weaker concentration can give a yellowish color, and it can take time for the color of brick red to be seen. Here you can see the experimental steps for testing for reducing sugar via the Benedict's solution. So just bear in mind the steps that are taken here. We are adding Benedict's solution to a piece of food in a test tube, and we heat that test tube in a water bath. Okay, so this heating in a water bath, okay, is important. Okay, so just be aware of that. Heating in a water bath. And here are the positive results. So once we have the presence of a reducing sugar identified, Benedict's solution changes through these colors here through to brick red. Now it may stop at, let's say, this point or this point, depending on the concentration of the sugar. So as you can see here, after heating, it may go through stages green, yellow, orange, red or brown, depending on how much glucose is present. So depending on how much reducing sugar is present, it may stop here or it may stop there. And a good positive test for a reducing sugar would be brick red. Okay, if there was no reaction, then the Benedict solution would just remain blue. Okay, and there's a bit more information here which you can take down, as it says sugars are classed as reducing sugars. Okay, and here it says Benedict solution is heated for a few minutes. This is important. Okay, so you can take this down in terms of the method of what's happening. Just note down what we're doing. We're taking Benedict solution in a test tube, heating it in a water bath for a few minutes. Okay, and testing for a reducing sugar. Now, a positive test gives a brick red color change, and here they've used the word precipitate, so you can use that word as well. It's a good word to use. Examiners like to see that word in the exam when you're using it, when you're explaining the color change. So you can say brick red precipitate, okay, with Benedict solution. Okay, and there's a bit more information on the precipitate here as well. Now, once you've summarized this in your own words, okay, so you've got your method at the top in terms of how we actually use the Benedict solution in a water bath and the different positive test results you will get. Once you've got a clear idea of that and a brief method summarized in your own words, I want you to again take a note of the safety precautions for this particular test. Again, similar to iodine, wear safety goggles. Benedict solution is an irritant, and avoid contact with skin and eyes. Main thing is understand that it's an irritant, so make sure that you're taking that down. Do not say it's corrosive. Okay, ensure that you understand how Benedict solution can be harmful. It's an irritant, and iodine is also an irritant. When we test for proteins, we use a solution known as a biuret solution. This solution, biuret, is blue in color. So, can you write down? We take a blue biuret solution. So, blue biuret solution. You can put reagent down or solution, it's fine. So blue biuret reagent or solution turns purple in the presence of protein. So turns purple if protein is present. So this is your test. Now you can have two forms of biuret, biuret A and biuret B. Now biuret A is essentially put on the first instance and into the test tube with the sample, and then you give it a small shake, and then you put biuret B into the same test tube and you give it a shake. Now, by doing this, you are looking for a positive color, purple, if protein is present. If protein is absent, the Biuret solution remains blue. Now, I'm going to show you a diagram and a method where Biuret A and B are explained in a bit more detail in terms of their properties and their chemical hazards. Here you can see the method for testing for proteins with Biuret solution, and you can see here to the method on the left, you can see where it says add 1 centimeter Biuret solution A to the food, and then you add 1 centimeter solution of Biuret B. So you can see that the Biuret is split up into two components. Now, what I want you to take note of is the method on the right, because that is from your exam board. And if you look carefully at point number 2 from the method, it's asking you to focus on, or it's put in bold, the risks associated with Biuret solution containing copper sulfate, which is poisonous, and sodium hydroxide, which is corrosive. So it's asking you to handle the solution with care and wash immediately if you spill it on your skin, and wipe up any spills because of the hazardous nature of the chemicals. So you are being alerted to a significant health, let's say harmful component to this test. So you may be assessed in your understanding of the hazards and precautionary steps needed to be taken when conducting the test for proteins. What I want you to be also aware of is the Biuret A, where it's corrosive, and if you read your method part 2 from your exam board, if you read the bold description again, you should be able to identify your corrosive substance. So what I want you to do is take a quick method, a summary method of the test for proteins, include a simple diagram, and note down any hazards that are involved, a brief hazard, maybe 2-3 lines, and precautionary steps that should be taken to avoid these hazards. The final test is for lipids, and this is going to be slightly different. I'm going to ask you to give your research findings for lipids. So you're going to carry out a research task for the test of lipids, and you're going to post it back on Teams. I'm going to give you a keyword, emulsion test. So using this word emulsion, so the emulsion test for lipids, I want you to try and find a simple method that is carried out to test for lipids via emulsion, the reagents involved, and the observable change. So a simple method, a simple diagram, and a few lines just outlining the hazards that are involved and any precautionary steps. So once you've found the answer for the emulsion test, can you post it back on Teams?